welcome everyone to today's webinar. So I'm Toby Schneider, and I'm happy to present my work that I did together with Ray and Isla at UMass during my postdoc time in 2019 to 2021, I believe. And I will take you on a journey to southern Greenland and talk today about tracing Holocene temperatures and human activities in Greenlandic lakes. However, I'm only talking about one lake today. I realize that only too late, but we used sediments from Lake 578 in the southern part of Greenland and combined hyperspectral imaging methods together with lipid biomarkers in order to reconstruct the Holocene lengths, anoxia, productivity, and temperature from this region. And of course, this work only has been possible with many people in a team, all the co-authors, but then also William Daniels, who helped us in the field, and also the reviewers of the paper, actually, that had really great feedbacks. And I don't know, probably one or the other would be around today too. So just to warm up. Yeah, let's see. To warm up, I show you this figure about recent global warming. And what we learn from this is basically that the polar regions warm at an increased speed compared to the global mean. And this is referred to as the polar amplification in the Arctic. It's the Arctic amplification. And this has a direct impact on the latitudinal temperature gradient, for example, influencing weather over Europe. So the heat wave in summer 2022 is partially attributed to this. And by the way, we just had the warmest August on record again this summer. And so these trends need to be set into a longer term perspective. And we can compare them, for example, to previous warm phases, such as the Holocene thermal maximum. And what we also see is that particularly Greenland is also affected. And of course, there are also many local direct impacts. For example, peripheral glaciers in Greenland, what we see here, that used to be all ice, and now there are entire lake and river systems on the glaciers, or the lakes are at the edge of the glacier crevasses, and also they're melting back rapidly. Of course, another impact would be the melting permafrost, bringing more nutrients into the system, dissolved organic carbon, for example. That has also a dramatic impact on freshwater ecosystems, such as, for example, lakes. And particularly in lakes, they're defined by dissolved oxygen availability in the bottom water, for example, and that's a key indicator of the aquatic ecosystem health. And so that plays a critical role, for example, for the entire ecosystem, but also for drinking water quality, but also about the biodiversity, entire nutrient cycles, or greenhouse gas emissions too. And influencing factors of this dissolved oxygen in the lake is basically some of those are physical, such as ice cover duration. For example, when the lake is ice shut, aeration cannot happen anymore, so atmospheric exchange cannot take place anymore. Or then the water temperature, such that the warmer temperature water can dissolve less or carry less oxygen, for example. But then there are also chemical and biological processes, such as, for example, respiration, that uses more oxygen, and or photosynthesis that would produce oxygen. And so in the Arctic, there's still a lack of data, or it's still challenging to have enough data. So, for example, on this map, we see different lakes around the Arctic Circle, and each of these dots, yellow, so that's a temperature anomaly map, by the way. And down here, please refer to this scale for deciphering the different colors. And each dot here represents either a time series of measurements, of monitoring measurements of dissolved oxygen, or single point profiles. And Clanton and colleagues, they nicely put together a literature review collecting all this data, and they had this nice time series highlighting how many lakes actually are being monitored for dissolved oxygen. And so what we see is that there's definitely a lack of data before the year 2000, and then the numbers are now slightly increasing. And also what we see is that there is a scattering, and our very heterogeneous coverage. So for example, Siberia hasn't that many lakes, and also Greenland, southern Greenland, for example, there's also a huge lack. So there's still limited data, and particularly also long-term measurements are lacking. For example, Holocene length reconstructions. And there are different proxies that we could use, for example, biological proxies such as assemblages of algae, or chemical proxies such as redox conditions that we can use to reproduce anoxic conditions, or now also hyperspectral imaging techniques that allow us to disentangle different pigments that are being produced by, for example, anoxic bacteria that thrive in anoxic conditions. And that's what we would use today as a proxy to reconstruct anoxic conditions, as well as also productivity of the past. And later, I will talk more about the technique. On the other hand, there's also still a lack of temperature data, particularly in the deglaciated areas. So the nice figure from Jason Breiner and colleagues that compiled all the different reconstructions available in 2016. So there are more being added now around Greenland, and also the archipelago from Canada and Labrador. And they compiled all these different reconstructions, and basically from different archives. So for example, ice cores, but also some tree rings, marine sediments, and also some lakes. And what they found is that actually the expression of this Holocene thermal maximum is rather scattered. So they ordered those along a latitudinal gradient. So this reconstruction up here, each line is a reconstruction, is in the north, from early Holocene to the late Holocene to the right. And they found a trend that it may be that the early Holocene, at the mid-Holocene thermal maximum faded out a little earlier in the northern part compared to the southern part, which would somewhat be expected, but there is still a heterogeneous pattern which can be attributed to different proxies in different archives, for example. So we still, particularly in the deglaciated area, we need to have some more data to constrain, for example, climate models, and also to understand what's going on in lakes. On the other hand, and our new or relatively new techniques, lipid biomarkers, for example, such as glycerol dankyl glycerol tetraeters, GDGTs, that can be used as powerful thermometers, and they are being employed in Greenland, but not that many reconstructions have been done so far. So that's the other proxy that we would use for temperature reconstruction. And now the general research questions of this project were basically how can we learn something from hyperspectral imaging and lipid biomarkers, and how can we combine them effectively? And then how did the Holocene variability look like in Lake 578? Particularly, how did anoxia and temperature and productivity co-vary? And whether we see differences in natural versus anthropogenic variability through the Holocene, and of course also how these reconstructions or the reconstruction from Lake 578 compares in a regional context. So Lake 578 is a relatively small lake. It's sited in southern Greenland, here in the eastern settlement of the Norse. The Norse arrived there by Eric the Red in roughly 985 CE. And that's actually the fjord here where they arrived, and they probably arrived up here somewhere. And as you can see, like 578 is this small orange area here. It's located at roughly 155 meters above modern sea level, and it's close by a sheep farm. And also there are some ruins of Norse settlements directly in the watershed. So these ruins are stables for sheep farming. And yes, the bathymetries on the lake is rather small. However, what's striking is this huge area. So the darker the color, the deeper the water column. And what's striking is this huge area of relatively shallow water. It's only 2 meters water depth. And so the Myriophyllum alterniflorum, this plant is really abundant in this area. That's typical for shallow water in relatively oligotrophic lake water. And also this transect, or this section of the lake, shows that actually the very shallow area here, so that's a water depth, is the largest fraction of this entire lake. The water column properties have been measured and monitored from Boyang, Ray, and Isla from 2016 through 2019. And so this is the temperature profiles, water temperature over time from 2016 to 2019. And each of these lines represent the terminal temperature logger that were deployed in the lake at different altitudes. So, for example, the dark blue one is the bottommost recorder, and the dark red one is the topmost recorder. And what we learn from this time series is basically that current state of the lake is a dimictic regime with long ice cover in November through May. So what we see here, starting in June or in the summer, and the topmost thermal logger is the warmest, and the bottommost is the coldest. And then during the autumn turnover, they line up because we have a mixed water column, so the temperature is evenly distributed. And then during the winter time, you actually have a inverted temperature reconstruction. So the water at the bottom is the most dense one, and the lighter water, the colder water, so that 3.98 degrees C, and the overlaying water is colder. That's why it's also lighter and or less dense, and that's why we have this inverted temperature stratification. And then in spring, after ice out, the entire thing mixes again, and then we have again a thermal stratification. They also took point measurements of different other parameters. Each line here represents a different year, and what we see in 2 years, and there's a nice thermocline here in the red color, for example, and the brown color. However, there are also years where it didn't occur to be a clear thermocline. And of most interest, of course, is the dissolved oxygen. What we learn here is that in the 2 times when they measured oxygen, that it actually was during the summer stratification, that it actually was distributed through the entire water column. That would indicate that at the moment the lake would still be doing okay in terms of dissolved oxygen. So Lake 578 has been subject to this large project from Ray and Isla, and particularly driven by Boyang Chao, who was the PhD student working on it. And so some work has emerged now. In 2021, that was the first study, which basically established the very first in situ calibration for these GDGT thermometers. And then in 2022, that was a study that, based on relative humidity reconstruction, could show that one aspect of the demise of the Norse actually could have been drier conditions. And then this study here is the one that I present today, which sets everything into a Holocene Holocene length context. And recently, there's also a palynological study that showed or zoomed into the Norse period. Now we took the sediment core for our work in 2019. The other sediment cores, they were from 2016, from Lake 578. And as the other cores already have been dated, and been published, we could do a cross-core correlation in order to project the age model from the 2016 core to the 2019 core. We did that based on visual inspection as well as with proxy comparison, for example, hyperspectral imaging. So these are the green pigments measured in the blue curve in the 2016 core and in orange in the 2019 core. And so we were looking for these easy identifiable features to connect the cores. And the same we did also for ITRAX measurements in current to current, for example, where we could find these matching points. So we could basically use or project some of the samples. However, we also added new dates and also new independent biomarkers. So the Norse, they brought to Greenland different species that were not native there, and they can be used as an indicator of when the Norse arrived. And one is Rumex acetosa, the other one Ranunculus, and sometimes also Coprophilus fungi that can be used to identify the arrival of the Norse, respectively of their sheep. And so we found a layer of these, or a clear onset layer in the sediment. However, it was slight discrepancy because in the originally published age-step model, so in the projected one to the 2019 core, this pollen layer actually fall roughly 400 years earlier. So instead of 985 or 990 when the Norse arrived, it dated to roughly 600 CE. So it was way too early. And this, of course, that would be a major discovery. However, we, of course, needed to corroborate that with additional dates. And so Ray went back to the sediment cores and picked some more samples, and particularly one layer with charcoal that was just next to the pollen layer. And we dated this terrestrial material. And unfortunately, we couldn't confirm that that the layer dates to 600 AD, but roughly to 950 CE. So actually, there was a age offset in the previous chronology that probably arose because of a reservoir effect. And so here, just to zoom in, that's the age model, the depth versus age. That's the early Holocene, and that's today. And if you zoom in, and each curve basically represents age model in orange or reddish colors, that's the uncorrected age model. And in black and green, that's the corrected age model with this new added char material sample. And if you zoom in just to the blue square, we see that actually there's this age offset between the two models. So, however, now the pollen layer, which is here roughly at 50 centimeters, dates to where it is expected when the Norse first arrived. So based on this investigation, we established our final age model, which is presented here in the middle, and also which takes into account the landnam, respectively the pollen layer. And also what we see is that the entire section is roughly 2 meters and dates back to roughly 11,000 years BP. Here on the left-hand side, we see a pictograph of the sediment properties and different fascias, 1, 2, 3, 4. And the first two fascias, they are very clay rich. Now, clay in this area, relatively close to the sea and also at relatively low elevation, that may point towards isostatic uplift of the deglaciation rebound from the ice sheet, meaning that could be an isolation lake that would be characterized by a clear contact. And so that's been done in the region. And these contacts usually are very clay rich. And so the question is, well, what's actually the ontogeny of this lake? And so a survey by Sparnbaum and colleagues, they investigated many lakes just in this area. So each point here is one of these lakes at different altitudes, and they used these transects of these lakes in order to reconstruct the relative sea level changes over the Holocene. And so the red dot here refers to Lake 578, which should be just through this small map. So it's just next to KA1 on this side. And what this Farnbaum found is the lower, sorry, in gray, that's the ocean. And here that's a lake very close to the ocean. Found that there's a clear contact in these lakes that are very close to the ocean. So there's a marine, they did that based on different biological proxies, assemblage and presence of certain species of microorganisms such as algae. And they, for example, found here in this lake K6 a marine species until a depth of roughly 380 centimeter. And thereafter, they saw a drastic shift towards limnic and terrestrial species. And so they identified that as an isolation contact. On the other hand, Lake K1, they did not find any evidence of marine species. So that was an indication for them that, okay, actually, this lake was already above the sea level in the early Holocene. So what we learned from this exercise is that basically Lake 578, which is even 50 meters higher compared to K1, is probably not a marine isolated lake, but the grain size distribution rather may suggest that it could be glacial water. So it's not a marine isolation basin, but rather probably a glacial, post-glacial lake, because a survey by Levy and colleagues 2020, they nicely delineated the regional deglaciation history, and they found, based on boulders and bedrock exposure dating, that in the Qaqortoq area, which is here, so that's South Greenland, and in purple, that's the Lake 578, they found that timing of the deglaciation was roughly between 12.5 thousand to 10.6 thousand years ago, which basically aligns with all the sample that we found in the Lake 578 record. And so in conclusion, that appears that Lake 578 is actually a terrestrial archive, a continuous archive through the Holocene that contains information about the environment and also probably temperature. And temperature, we measured based on the GDGTs that I've already mentioned, and we used a specific subset of GDGTs, the so-called branch GDGTs, and they're characterized by number of methylations of these minibars here and number of cycles, these cycles here. And that's basically also the notation or denotation. So one refers to the tetramethyl, means that's like 4 of these bars, and ABC to the number of cycles. And there are 3 different types that we investigate. In total, there are 15 compounds. So pentamethyl with 5 of these methylations and hexamethyl with 6 of these methylations, and then each has also isomers on for these two compounds here. However, now we have this data set, and in order to get a temperature signal, we need to extract that using different calibrations. And these calibrations are usually established using environmental gradients in different lakes or soils, and to extract the composition using different linear regression models, for example. And so that's why there are numerous calibrations being published. And so it's always tricky to find the right calibration, also to calibrate all of that without making any errors. And during the course of this project, we also started developing software that basically calculates all the different indices and different calibration, over 100 so far. And we implemented that in R, and over the course of the project, it got larger and larger and larger. And at the moment, we are just finalizing a publication to finally publish this software. And so people of you who work with GDGTs probably watch out for this paper or contact me for the software, etc. So coming back to the Lake 578 and temperature reconstruction, we basically applied two different temperature reconstruction. One is global, incorporating global data set, although I have to say global because we need to be careful, because as we see, the poles are roughly, there are hardly any samples for the calibration set. So there's really a lack of data still, and it's more centered around the mid latitudes and probably the equator. And on the other hand, the very first in situ lake calibration established by Boyang in Lake 578. And these are the two calibrations that we use. And that's what we see here. So we calibrated our branch GDGT reconstructions over the Holocene. Here's the early Holocene, and here's today, basically. And what we see in black, that's the summer mean water temperature reconstruction based on the in situ calibration. And these colored ones are the so-called month above freezing calibrations. So probably a side note here, the summer mean water temperature incorporates the temperature of June, July, and August. And the month above freezing, all the temperature that the mean temperatures, or the month with the mean temperature above 0. So a much larger season. And what we learn from this is that there's definitely a gap in absolute temperatures. So the in situ temperature reconstruction is much warmer, but that can probably be attributed to seasonality. And then there's also much larger root mean squared errors in these reconstructions here, and that's due to the many different samples. And also, we need to keep in mind that for these global data sets, there are different sample types included, for example, also soils, so not only lake samples. And that's why we mostly rely on the summer mean water temperature. That's the best we can do in this lake. And these, if you wonder about these two gaps here, these are not hiatus from the sediments, but these are rather caused by COVID. So there was a lab shutdown, and unfortunately the project ended through this time. And so we could not finish these two sections. So that's a simple and unfortunately a simple explanation for these gaps. Just keep that in mind, because for example here we have now the anomalies. So it's the same temperature reconstruction from based on the in situ temperature, but calculated anomalies over the Holocene. But for example, we see now that here it's just linearly interpolated. So we need to be careful between these two periods, because we basically don't have any data. But what we see now from this reconstruction is that Lake 578 had relatively cold anomalies in the early Holocene, which is expected, and roughly at 9,000 years BP starts to get more positive anomalies. So, which then probably could be at some point the Holocene thermal maximum, and then roughly 1,200 years there's a strong cooling trend towards the top. And what we also see are these relatively rapid cooling events. And of course, if you think about southern Greenland and the vicinity towards the Labrador Sea and the ocean, we think about rapid freshwater discharges caused by the Laurentide ice sheet deglaciation, for example. And so we compiled basically 4 different stacks of these events that were reported, and just to see whether or not we see actually influence. And for example, here it may very well be that we see some direct influence of these freshwater inputs in the temperature reconstruction from Lake 578. If we set this temperature reconstruction now in a larger context, regional context, we can compare it to different other reconstruction. On top, we still have the anomalies of Lake 578, and then ice core stack by Kobashi and colleagues. And then in the middle, these are all temperatures anomalies. So we find the time here from early Holocene through late Holocene. And then the hemispheric reconstruction by Market and colleagues, including the records from 30 to 90 degrees north. And then the same for Darrell's group, Kaufman 2020, but only focusing on the reconstruction 60 to 90 degree north. And finally, the reconstruction based on pollen reconstructions and by Gajewski, 2015, for just 4,000 Greenland. And what we see here is basically that compared to the other reconstructions, this early Holocene temperature increase seems to be relatively delayed in Lake 578. So others start deviating from the zero line earlier compared to Lake 578, and also that there's some probably higher variability in Lake 578, and this warm phase seems to be also rather longer compared to the more regional and hemispheric reconstruction. And one special feature is this cooling trend here, which is contradicting towards the others that show actually the contemporary warming, which we also would expect to see. However, Boyang nicely documented in his nice paper in 21 that this may be actually an artifact of the GDGT temperature proxy. But does this suggest that Lake 578, as we heard last week from Willem van der Bilt, that actually the lake temperature is decoupled from the atmospheric temperature evolution around this area, and to further explore this, we set it into a different context also and compared the reconstruction to previously multi-proxy reconstruction that takes into account the North Atlantic Fennoscandian stack. So all of these, so that's the North Atlantic, that's Iceland here, that's and here it's all these dots and numbers are reconstructions that have been incorporated into this stack. And basically what they saw is that their PC1 of their analysis nicely follows the insolation trend. So that's early Holocene to late Holocene, and the black curve is the insolation as reconstructed. And the red line is the summer temperature of based or derived of this multi-proxy reconstruction, and nicely follows this trend. Another feature they found was PC2, that this follows basically the model of the Laurentide ice sheet. So it's the black curve, and nicely follows this one. And that suggests that actually the Laurentide ice sheet probably had a direct influence in this region, on the climate in this region, due to the changes in topography, albedo, and meltwater discharge. And Renson and colleagues in 2010, they investigated this exactly based on these assumptions. And what they found is, when they compared the year to 9000 to present, so it's exactly the timing of when Lake 578 starts to show warmer temperatures. What they found is, so here's Greenland, that's the Labrador Sea here, and that's Iceland, and over here's Scandinavia. And what they found is a blob of cold anomalies. So the darker the color, the colder the anomaly at exactly this time, suggesting that this is a cold area here, and is distinct from the entire atmosphere, so that which would make sense that we would see different temperatures compared to the reconstruction that incorporated the entire atmosphere. And that's exactly what they did. They found with their reconstruction some regional differences. So in yellow, we see again this market reconstruction that we saw before, just the other way around plotted. And in black, here it's a reconstruction that incorporates the global multi-proxy temperature. And in blue, most interesting, is now the North Atlantic Fennoscandian stack. And what we see is that this delay, or this the warming trend, is also delayed from the Fennoscandian stack compared to, for example, Marco reconstruction. And of course, now is the question, how does Lake 578 match with this? And so we plotted that on top of it in green, and we see basically that it follows nicely the NAFS stack until roughly 5,000 years BP, when they start to decouple between the NAFS stack, which goes towards colder temperatures here, and Lake 578 stays relatively warm. However, again, we have a data gap here and here, which is unfortunate for this situation. But at least this may suggest that no, Lake 578 is actually not decoupled from the atmospheric temperature, but rather that this area is not representative of the entire hemispheric temperature evolution at around this time. And now we go towards the anoxia and productivity reconstruction. And just for the concept, so we have Lake 578. And when we, for example, where we have mixes and a well-mixed system, and if we put ice cover on top of it, the oxygen starts to being consumed by different microorganisms, and at some point that's the ideal setup for methanogens and different bacteria that can live there, and they start producing chloropigments, and when they die, they get settled into the sediments. And then similarly, if we have productivity and well-mixed system, for example, algae or diatoms can produce green chloropigments, and when they die, they also get settled into the lake. So we take this sediment, and we can now use hyperspectral techniques to deciphering these different pigments, and that's based on a non-destructive method. We basically have a pixel size of 70 micrometers. That's the scanner up here on the right corner, and we scan the entire sediment core, and per pixel we measure roughly 600 nanometers of wavelength. That gives us a spectrum. For each pixel we receive such a spectrum, and the green chlorophyll or green pigments, they absorb at a specific wavelength, 670 nanometers, represented here, and the bacterial chlorophylls absorb at relatively, probably at around 850 nanometers. And these troughs are directly linked to the content. So the more green pigments we have, the deeper this trough gets. And we can calculate this trough depth, so-called relative absorption band depth, and use that to quantify the green pigments, which are indicative of productivity, respectively the same for bacterial pigments for anoxia. And then we get a spectral map where we basically see the darker green contains more pigments, for example, compared to the lighter green. And this is what we did for Lake 578. Here in green is the distribution of the green pigments from modern times to all times. And in purple, the same for bacterial pheophytine. And in the middle is a true color image. We calculated then based on the mass accumulation rate, pigment fluxes, and subjected those to different statistical treatment to calculate or identify periods of significant change. And we see here on top the green pigment fluxes from early Holocene on the right towards today, in the middle anoxia, and on the bottom the mass accumulation rate. And we followed the simple workflow from Simpson 2018 that uses general additive models, and that allowed us to calculate different confidence intervals and take the first derivatives of each time series, and basically in order to detect significant changes. And basically when the first derivative deviates from the zero line with the confidence interval of 95%, then that's a period of significant change. And like this, we could easily identify these different phases. And so per time series, we found multiple changes. For example, productivity, I found particularly here around the Norse period, which is highlighted by this gray bar. And in the recent time, we found significant changes so that we have more compared to the natural background. And for anoxia, we have several phases in the early Holocene and also one around the Norse period. And for mass accumulation rate, we mostly see that in the early Holocene. So to put everything in a bigger picture, and what does that actually mean? I plot here a synthesis. First, that's plotted on H now. Lithogenic fractions based on XRF and dry bulk density and CT scan here, from early Holocene to late Holocene. And then the productivity, all the green pigments from hyperspectral imaging here. And from the lipid biomarkers, we also calculated the total lipid extracts, and that also nicely seems to represent the productivity. And then the same for anoxia. Here first with the spectral derived pigments. And interestingly, we also found proxy very useful from the GDGTs, two specific compound GDGT0 to crenarchiol, which also represent anoxia. And these two curves, they are completely independent and they really nicely match the story. And thirdly, also from the eye-tracks measurements for sulfur, where we also see that somehow it seems to follow a little bit this pattern, which could be indicative of more methanogenic production. And then finally, the temperature reconstruction. Cluster analysis revealed 4 different clusters. And for example, here the early Holocene is characterized by relatively low summer mean water temperatures, which we see here, and a high mineralogic content, which could be due to the glacial input and low productivity, and also relatively high anoxia. And now the question, of course, is what could be the driving factor for this anoxia? And so we came up with 4 different hypotheses. One is the ice cover that we saw at the beginning, so the lake is ice shut, and then the oxygen is being respired in the bottom. Could be possible that this is a valid hypothesis, and then seasonality, so that extreme summer productivity and extreme ice cover in winter could increase the or decrease the dissolved oxygen. That's also possible. And then the wind system, so that actually the wind was shut, so that the lake couldn't mix anymore. However, that's tricky to show because there are hardly any models that are providing the right resolution to test this hypothesis. And then another factor could be is actually the thermometer not correct, and so it's actually anoxia influencing the thermometer, and this seems to be less likely because the temperature reconstruction seems to follow nearby reconstructions too that were not measured with GDGTs. And so we think that mostly the three first hypotheses would be possible. And that means that also here, when we see in the second phase of the early Holocene transition towards mid-Holocene, when we see a rise in summer mean water temperature and a slow decrease in anoxia, could actually mean that we transitioned from an ice-covered lake to a lake that started to mix again and distribute oxygen. And the mid-Holocene may be characterized by a dimictic system highlighted by the decrease of the anoxia to zero, basically, and also start of the productivity. And then finally, the most interesting phase is the anthropogenic pressure phase. Here we see the surge in productivity in the last cluster, along with the anoxia that we saw before, even though they're lower temperatures. And so, and in addition to this, we also saw increase in pollen of this Myophyllum alterniflorum species, suggesting that there could have been a strong lake level change. And that's possibly due to artificial damming, because in neighboring areas, they found during archaeological surveys that they actually altered, Norse people actually altered lake levels, so that actually the lake was probably very small, and then they started flooding it, and that would also explain this large area of relatively shallow water. So to sum up, what did we learn from this study? This study basically suggests that the method combination of hyperspectral imaging and lipid biomarkers is very powerful to disentangle different mechanisms driving aquatic ecosystems and temperature. And then also what we learned is that for the GDGT thermometry in the northern parts of the world, that there's still a lack of training sets for calibrations, and also that it's really important and crucial to use a multi-dating approach to date these lake sediments, because sometimes it seems to be the most ideal age model, but then independent chronomarkers can basically throw over the entire thing, so that one should always also combine different methods. And in terms of science, we basically learned that there are strong natural and anthropogenic shifts in this lake, particularly for anoxia. So when it was in the early Holocene, probably caused due to cold inverse stratification or strong seasonality and ice cover. It was in the late Holocene anthropogenically induced, so means increase of productivity, decrease of oxygen in the system, and probably in the future, according to the models and the regional or other Arctic monitorings, it may be that it actually could evolve into a third phase of anoxia, namely the warm temperature stratification, but that's just speculation. And finally, what we also learned is that the Norse impact seemed to have altered the the entire lake system in a way that it did not go back to the pre-Norse, pre-anthropogenic lake condition anymore. So that is really a long-lasting effect. And with this, I would like to thank you very much for your attention, and sorry if I was a little long.
Thank you, Tobias. That was really interesting. We're going to open it up for questions. Anybody on the chat or in classrooms?
Hello, I think we have a question here in Bern.
Nice.
Hi, thanks for your talk. Can you hear me?
Yes. Sorry. Yeah, I can hear you. Sorry. Yeah, it took me a while, but yes, I can hear you. Sorry.
Okay, thanks. If you go back to the synthesis with all the proxies, then during the human period, the period where the Norse people arrived, there was a high increase in productivity and anoxia conditions. And then later, when they left again and the modern settlers arrived, then the productivity seems to increase again, but there is no anoxic conditions. What is the explanation for that?
Yes, that's a very good point and very, very careful observation, I would say. Thank you for this. So, no, it's actually also, we also wondered what that could be. And one explanation is, so here it happened this probably artificial damming leading to increased nutrient input and also increased productivity. And due to the setup and direct impact really in the watershed, could have basically led to much more consumption of oxygen. So we also see that it's slightly delayed, the anoxia signal is slightly delayed. So first you have huge productivity and then delays. And so why is that not the case now? Maybe that indeed the bacterial community changed due to different introduced other species, or who knows, and so that they actually are not producing these pigments anymore. So that could be one explanation, because we rely on this, right? Like we need to have a producer of these pigments if we want to reconstruct this anoxia. And we actually see in the other proxy, we see somewhat probably indication that it could be still anoxic based on this other proxy here. However, that's not really clear. Another explanation could be that it has not yet arrived, so that sort of the system got into equilibrium also with turnover again, and that actually probably also the ice cover during this period was longer compared to today, and that it's now due to shorter ice cover period actually that the lake is stratification is too short to actually establish a huge anoxic condition in the bottom water.
Good, thank you.
Thank you.
I'm wondering if you'll ever get a chance to run the samples. I mean, it's just really interesting to see how much they're sort of decoupling during that from the Laurentide stack. Is that something on the horizon?
Yes. No, unfortunately not. I would love to do though, and probably I have to chat with Isla at some point. I don't know whether she is also around, but so that we could fix this, because yes, totally right. It would be super interesting, and it's also really frustrating because we spent so much time and we couldn't finish. Yeah, the last step basically. Yeah. We couldn't do that. Yeah.
What's the rationale behind artificially increasing lake level?
Sorry, I didn't get the first part.
What is the reason that the North
Yeah.
increased the lake level?
That's a very good That's a very good question, and I would like to refer you to the paper from Sascha Krüger, who established palynology in this lake. So basically, there have been, so they used to drain. Sometimes they used to drain some swamps, even so they were they used to be sophisticated water engineers, I would say, and sometimes they tried to dam some basins to actually have more freshwater supply. And so here, honestly, I don't know. Probably it could be that there's also a mill involved, so that they even would have some more sophisticated damming to do. But that's I think that's also more speculative, also in the other paper. But there have been ruins, not exactly in this watershed, but further away, that would indicate such things.
Well, thanks.
Hi, Toby. This is Elizabeth
Hi.
in Buffalo. Thanks for the awesome record and the nice talk. I'm curious, maybe more just speculation from all of our parts as organic geochemists, but I'm curious if you have thoughts about why this record, for example, would not, the GDGT inferred temperature would not be influenced by anoxia, whereas in other records it might be.
That's a very good question. No, I cannot answer this one. Actually, I think we actually see, if we plot all the different compounds, we see some differences in the very earliest samples compared to the others. However, that's only the very bottom part. And then others during this anoxic phase still seem to match up exactly with the temperature calibration set. And so the temperature calibration set is, basically produced during, okay, that doesn't make any sense, during oxic conditions. So it probably is a little contradicting. No, I don't know exactly why, but it's just comparing it to the reconstructions from Igaliku and one other lake in the vicinity, that they use different proxies. It seems just to explain the same pattern. That's why we claim that it seems not to be affected by anoxia. And we also have to, I mean, GDGTs are still understudied. I know there are now the community switches now also to laboratory experiments, in vivo experiments, in vitro experiments, exactly exploring different conditions and what's the effect actually on these compounds. And probably it's not yet really deciphered.
Thanks.
With any further questions from Bern, from UMass? Anybody online? Okay, I want to thank you so much for the really interesting talk, and remind folks that we have another webinar next week on Wednesday on Tancred Ledger from Sheffield, speaking about calibrating paleo ice sheet model simulations. So we look forward to that. See you next week.
Thank you so much. Have a nice evening, day, afternoon.
Bye.